Great. So welcome everyone to this month's Limnoseries. I'm Lisa, based in Switzerland. I will be your host today. And I will start by briefly introducing the GSA and our speakers today, Mridul and Ravi. So quickly, GLION is a global network that gathers scientists from all around the world working on immunology in order to conduct research on international team science and advance our understanding of lakes. I am saying this because I believe that today there are people that are not yet in the GLION network. So if you do not know us yet, do not hesitate to visit our website. We also have an upcoming virtual meeting, so it would be a nice way to join the network for those of you who would be interested. And today, this Limno series is organized by the GSA, so the Student Association of this network. And the GSA's mission is to inform, train, and mentor students to allow them to participate in collaborative science internationally and also interdisciplinary. And here you can see our wonderful leaders, Lipa, Anna, that is also here with us, taking care of the technicalities of the Zoom, and Carthena. And they are also advised by Bas Iblings. So here we have different committees in the GSA. We have the workshop committee, so you can see the people involved in this committee here on the slide. So Kati, Jenna, Anna, Tana, Lorena, Carthena, Lipa, and everyone else. So this committee is taking care of organizing the Limnoseries like today. We also have a communication committee that is taking care of our Slack channel for Glion students that host seminars, also create content for the Glion website, like blog posts, flyers, etc. And we finally have a poster committee that is in charge of preparing and planning posters during the Glion meetings, for instance. So we are taking those few minutes to present you the GSA because we would really like to welcome as many students as possible to join us and take part in those committees. So it would be a great opportunity for you to get more involved in the GLIA network, to learn some new skills, meet new people. So do not hesitate to reach out to us after this sermon series if you would be interested. We are also looking for speakers for the next Limno series. We already have some that would be incoming, especially next month on grant writing, if some of you would be interested. So again, do not hesitate to reach out to us. It can be any kind of format, a workshop like today, or lecture, presenting your latest research. And finally, those Limno series can be found on our Glion website and also on a YouTube channel. So do not also hesitate to check those out. And now I would like to introduce our speakers today. So we have Mridul Thomas, which is a senior researcher at the University of Geneva in Switzerland. He got his PhD in Michigan University and then pursued with a postdoc at EVAG in Switzerland, in Zurich, and got a Marie Curie Fellowship at the Danish Technical University. And right now he's doing research on phytoplankton response to abiotic environment with a focus on temperature interactions and other drivers to determine the phytoplankton dynamics. He is also interested in connecting experimental and observational data with mathematical models in order to improve experimental design and data analysis in model-relevant experiments, which is why we are having this wonderful workshop today. And we also have Ravi Ranjan with us today, postdoctoral associate at the University of Texas in the USA. He also got his PhD at Michigan State University and pursued with a postdoc at Helmholtz Institute of Functional Marine Biology in Germany. And now he's doing research on developing mathematical models to understand primary producer responses to global changing and to develop informative designs for experiments to complement models. So I will gladly give you both the floor now, Mridul and Ravi. I think we're a lot of people today and we're all very excited to participate in this workshop. Thank you for being here today and giving us to us.
Thank you, Lisa, Anna, and thank you all for joining today. We're excited to see so many of you here. All right, can everyone see? Okay, so A long time ago, I actually used to do experiments, and I was rather terrible at them. But I also was extremely annoyed by this frustrating problem of how, once you decide to do an experiment to answer a question, how you actually figure out how that experiment gets done. And it seemed to me that there was a lot of lore and rules of thumb. And that lore and rules of thumb may work okay in a bunch of situations, but there had to be a better way of going about doing things. And that stimulated a long-standing interest in this. And so this is something that Ravi and I have been thinking about for a while, and we're excited to share some of our ideas with you. So before we jump into the work, I want to say that there's a few things I want you to take from this workshop. The most important thing you can do is to define a clear, achievable, and useful objective. Before anything else, you need to have that. This seems obvious, but I'm sure you'll all agree that there's plenty of work out there which doesn't seem to obviously address a clear goal. Consider what parameter or quantity is most useful to estimate and what level of precision you need. This will become more clear during the workshop. Think carefully about the statistical model, that is the function you are fitting, and also the sources and magnitude of error in your experiments. And what we will actually do today is what I hope you will do in the future, which is when planning your experiment, simulate data from your model and compare the precision of the parameters that you estimate under different designs well before you actually do the experiment. And finally, once you have this sort of workflow, it becomes easy and useful to check how robust your design is by changing different assumptions and resimulating. Now, have some work that we will do together and some work you all will do individually and in groups during these 90 minutes. We have sent you 9 R scripts. We obviously will not get to all of them, but some of the topics we talk about here and some of the material that we present, you will be able to work through yourself through those scripts. Okay, so to be complete, to avoid ambiguity, let's define some terminology. So an experimental unit is the lowest level that you manipulate in your experiment. A level or a treatment is the value of the driver or the factor that you use in the experiment. The design space is all the possible levels that you could use, that you choose from. And finally, replicates, of course, are experimental units that are subjected to identical levels. So suppose you're interested in how, say, a toxin affects a fish species. Your experimental unit will hopefully be an individual fish. The level or treatment will be the specific toxin concentrations that you use. The design space will be the number of possible toxin concentrations that you could use. And your replicates would be fish that are given identical toxin levels. Okay. With this background, we'll jump into the first topic people usually encounter when they hear about planning an experiment. That is classical power analysis, which is usually introduced in terms of an ANOVA. So an ANOVA is a linear model which you use to tell if the means of 2 or more groups are different from each other. So in order to do a power analysis, what you need is the intended sample size per group, the probability of incorrectly rejecting the null hypothesis, that is, when it is true. So this is a false positive. The standard deviation within the groups, and most importantly, the smallest effect size you are interested in. Not the effect size that you expect, but the smallest one that is interesting or meaningful. So with this as input and some maths that I will not get into, what you will get out is the probability of correctly rejecting the null hypothesis when the null hypothesis is false. That is what we call power. Now, As your model gets more complex, now the maths of a power analysis for an ANOVA with two groups is quite simple. But as your statistical model and your intended analysis becomes more complicated, power analysis can still be done, but the maths behind it becomes far more complicated. Simulations are an alternative that are slower and not as perhaps mathematically elegant, but their advantages are that they are easy, they're flexible, and they're really, really powerful. It's just 4 steps. You decide on the statistical model that you intend to fit to the data. You make up some numbers from the parameters for the parameters. You run it 1,000 or more times, and you calculate the percentage of times that you get a significant p-value. That is the power. So if you run a simulation-based power analysis and you see how, for example, on the x-axis here, we have sample size per group, and on the y-axis, you have power. We have set the other 3 numbers here. We've set the probability of incorrectly rejecting the null hypothesis at 0.05. We've set the within group standard deviation at 1, and the smallest effect size, that is the difference between groups that we're interested in, at 0.5. With those assumptions, with a simulation, you can see that you reach an 80% power at around a group size of 60, a 90% power at around 80. And then as you add more, you don't really get a whole lot more power. And on the left side, this is the same plot, but with the analytical result, which is the solid line here. And you can see it matches up with the simulation-based result quite well, which shows that a simulation gives you all the information you need. Now, on the right side, we have fixed the number of the sample size per group, but we have changed the amount of error. And you can see that after the error increases to about 0.4, suddenly you have a big drop in power. Okay. Now, there are a couple of scripts that let you work through this, but partly why I have not gone into details here is that power analysis focuses on the wrong thing. I do not believe it is particularly useful. For almost any experiment that you would consider doing, we probably know that the true value is different from 0. So finding a statistically significant result, which is what a power analysis tells you how to go about doing, is just about using enough experimental units. To me, this seems like a bit of a mathematical game. It's a dead end. If you use enough units, you will find a significant difference, but there's nothing more that we particularly learn from this exercise. So what should we be trying to do instead? Well, you can use the very same simulations, but not pay attention to the significance and instead focus on the precision. You can think about doing what you might call a precision analysis or precision planning. What do I mean? So let's go back to the simulation-based analysis. And as I said, when you start, you think about the model that you are trying to fit. Now, the standard model that you would fit when comparing the means of two groups is simply this. You see y is equal to alpha plus beta x plus epsilon. In case you're unfamiliar with this, this is just the equation of a straight line, a plus bx or mx plus c. And this epsilon is the error. So it is the noise that we get that arise out of natural variation and measurement error. So y, you have a response. Alpha is basically the mean of the first group. Beta is the difference between the groups. X is just a 0 or a 1, which determines which group which group's mean we're interested in. And then this noise term. So in order to do a simulation-based precision analysis, you can choose values for basically the mean of group 1, the difference between groups, and the standard deviation of this noise term. Now the mean is going to be 0, of course, because it's noise. But The standard deviation is what tells us how variable the data is, how noisy, how noisy the data is, and ultimately this will affect how uncertain our estimates are. Now, typically, remember epsilon is a random error, and typically it is normally distributed. Everything we're going to talk about today is going to assume normal or Gaussian errors, and but it doesn't matter if you want to explore some other kind. So you choose values for alpha, beta, and the standard deviation of epsilon. You choose the sample size per group. You simulate data 1,000 times. For each of those 1,000 times, you fit your model and estimate the alpha and beta. And then you can ask, how much variation is there in alpha and beta across my simulated data? And what you're going to see on the left is a sequence of different randomly generated data sets. And on the right is going to be a, a cumulative counter of the parameters that values that we estimate. So each of these simulations is random data which obeys the same parameters. And we can see that they vary in the estimated differences between the groups, the beta that we are interested in. I'm going to stop this now, but you can run this for as long as you like, and eventually it'll provide a nice, beautiful Gaussian distribution here. Now, that distribution tells you about how precisely you can estimate this parameter, in this case, the difference between groups, given what we have assumed, which is a particular sample size and also a certain amount of noise. Now, the standard deviation of this distribution is what we typically call the standard error. So this expected standard error is 0.37. But now over here, I've done this with just 15 individuals per group. You still get some random simulations where your estimates are negative, though they're quite rare. But broadly, most of the values are between about 0.3 and 1.6. But how might this precision change with sample size? Okay, so now I'm going to switch to a little demo in R. You have the script with you, but I'm just going to run through it over here. I'm not going to explain this in much detail to save time, but if there are questions about it that you want to send us in chat or send us an email later, you're welcome to. So we're going to load some required libraries, and we're going to set some simulation parameters. So delta here is the difference between groups that we're assuming. That's 0.5. And sigma here is the standard deviation of epsilon. So it's what's the standard deviation within group that we want to occur. So now we are going to define some numbers. So the number of individuals per group, the number of simulations, and now we're just going to repeat this simulation many times with the same parameters, but different random noise. And that finishes very quickly. And this is the plot you just saw. As for how the standard error changes with sample size, now we're going to define a range of sample sizes from 10 to 200 in intervals of 10. We're going to do fewer simulations so that this runs quickly. And we're going to save the standard errors, which is the standard deviation of this distribution. Okay. And this is what we get. Now, the mean standard error at any one sample size is basically telling us, on average, how uncertain, how variable are our estimates going to be for that sample size. So what you're seeing is that, of course, a sample size of 10 per group, you have a standard error of about 0.45. And adding just a few individuals very quickly reduces your standard error. So your precision increases extremely fast at the beginning. But at some point, as you add more individuals per group, you don't really gain all that much. I believe this is a power law. So this is a way to explore at what point you stop really benefiting from putting in more experimental effort. Okay, now we're going to switch back to this. Well, so what if, though, in this case, we varied the sample size per group, but we assumed a fixed error, a fixed standard deviation. So here's a little activity for you. We're going to spend 5 minutes on this using R script number 4, which you've received in your email. Figure out how the precision, that is the standard error, changes with this sigma, which is the noise that you are introducing. Keep the number of points constant. So I'm going to stop sharing and I'm going to let you all switch to that.
And if anyone did not receive the R scripts in their email, just message me on the Zoom and I can send them to you.
So the script already contains phone numbers, but you should change them to anything that you think might be plausible or interesting for your study system. So what I hope you would have seen is that depending on the error that you assumed, that you could get hugely different precisions. If you're doing an experiment with a sigma value of, say, 5 or 10, even though the true value, which is represented by this red line, is here, you could quite plausibly get extremely different values. So if you live in a world where you have this kind of standard deviation, then this sort of sample size is not going to cut it in order to get a precise estimate. So you can play with both these numbers. This is going to be measured, the sigma is mostly going to be determined by your ability to measure things precisely experimentally, and also the natural variability that you have. But the sample size is the main thing that you get to control. And you can explore these two to see how that will affect how precise and reliable your ultimate measurement and estimate is. Now, precision is good, but what are we actually trying to achieve here? Now, we often think that we are testing a scientific hypothesis or establishing causation in science, but there's other things that we might think are useful. We might do an experiment to predict something of importance. We might estimate an input for a mathematical model, which could then be used for understanding our prediction. We could also simply use an experiment to explore how some change affects some property of interest. Since it's most common for us in science to talk about testing hypotheses, I think this is important to talk about. What is a hypothesis? There's two different definitions we work with. And I'm going to say, in science, to us, a hypothesis is an explanation that relates to phenomena via some mechanism, and that mechanism is important. Not everyone will agree with this, but this is one that we believe in. In statistics, however, it's a mathematical statement that can be evaluated with data, often in a true-false fashion. Now, there has been a huge amount of confusion in science by this completely different and definition of hypothesis that comes from statistics. So let's talk about what a scientific hypothesis would be, the kind of thing that we would have to do in order to test a scientific hypothesis. So let's start with a general claim. Say biodiversity is related to the environment. And now we want to try and refine it because, as I said, a scientific hypothesis must contain a mechanism and must also be as precise and focused as possible. So we can refine this to the number of species in an ecosystem changes with temperature. We might say, really, it's the number of species per unit area, and also not just changes with temperature, but increases with temperature. We've added direction here. Now we might say the number of species per unit area increases with temperature because higher temperatures cause individuals to grow faster. Now we've added a mechanism. And finally, we might say the number of species per unit area increases with temperature with a slope on a log-log plot of 3/4 because metabolic rates increase with temperature with the same slope. And higher metabolism means more individuals are born, and more individuals that means that more diversity can survive. Now, we would call number 4 and number 5 here scientific hypotheses because embedded in here is a mechanism. But I want to note that you would use probably the same statistical model for number 3 onwards here, possibly even number 2. So with very different levels of question or hypothesis, the statistical analysis could be quite similar. But it's not just the statistical analysis. We might use different experimental designs for 2, 3, 4, and 5. For example, for 2 and 3, since we're just saying that something changes with temperature, we might just say, let's have 2 temperatures and we just measure the number of species. That's it. That gives us the answer to these two. But for number 4, we might, because there's a mechanism there, we need to investigate that mechanism as well. We not just measure the number of species, but we should also measure the individual growth rate. Because if the pattern exists, but the mechanism is wrong, we've learned something about this hypothesis that it's incorrect. And finally, for the 5th one, again, we would have to manipulate more than 2 temperatures here, ideally, because we have a specific slope that we're interested in. We'd have to measure the number of species and the metabolic rate and the birth rate and actually also the number of individuals. And ideally, we would actually want to manipulate the metabolic and birth rates, if that were possible, to make sure that we are not getting that slope for some other reason. Now, here's another group activity. Well, here is a group activity for y'all to spend 5 minutes on. Please take a screenshot of this right now. What I want you to do is pick one of these 3 questions below, or one of these 3 statements below, refine it as much as possible, just as we did. Design an experiment to test or answer it. What experimental levels will you use? If you can, even how many experimental units would you use at each level? Just have one person type out your answer and we'll have you share it in chat later. And we're just going to spend 5 minutes on this starting now, if Anna can send you all into small groups. Could you have one person from each group just chuck your final question into the chat? Okay, we've got one group that has worked on the looks like a biodiversity ecosystem function question. And they've got how does the number of species per unit earth area affect the amount of plant biomass in a grassland produced per year, because more species cover more soil volume. So again, they're thinking carefully here about the mechanism, which is nice. And as you make these questions more precise, it also makes your assumptions and the connections between different processes more clear. And hopefully that will also make clear to you the the side analyses and the additional assumptions that you need to investigate in order to properly test your hypothesis. So I am going to move on since I don't see any others yet. Okay.
Another one just came, I think.
Thanks. So I hope you would have recognized that it's not easy to make a question or a hypothesis precise enough to test well. So it is possible, and if you think carefully about it, you can do it. But testing rigorously a scientific hypothesis in a single study is rare. It's also quite rare to do a study which alone allows you to predict something useful about the world. And this is because the world is complex. We need mathematical models often to understand and predict how different processes interact and affect some larger property that we're interested in. It is very complex mathematical models that we rely on ultimately to predict things like future CO2 levels and biogeochemical cycling. But even much simpler mathematical models to investigate questions like, can these two species coexist if the amount of nutrients in this lake increases? Precisely estimating the parameters for these mathematical models is a valuable goal for experiments, and one which I think ought to be, if not the main focus, one of the main foci. I also want to note that if your focus is on precisely estimating a parameter, those precise estimates will also help you with prediction and exploration, and often even testing hypotheses, though that may happen via a meta-analysis. So I think it's worth thinking about the fact that ultimately much of what we want to know requires a mathematical model to investigate carefully. Because of that, and because an experiment often cannot give us the answer directly, we need to think about experiments often as providing input for mathematical models. So what kind of experiments are useful for mathematical models? This really depends on your question and your system. So do not take this as in any way rigorous or definitive, but these are some general pointers. If you are dealing with a continuous variable, like temperature or nutrients or diversity, regression is more useful than ANOVA, so continuous gradients. Even when you're comparing two groups of species, consider comparing their model parameters. What do I mean? For example, comparing their feeding rates. This could tell you something about how they perform in an ecosystem. But things like those feeding rates arise from an experiment that has a continuous gradient. Understand the appropriate equation or function that applies to what you are trying to understudy. Is it a straight line? Is it saturating? Is it unimodal? What are the parameters of this function? Think about the level of precision of those parameters. Would a 20% error matter a great deal for the kind of predictions you want to make for the mathematical model? What about 50%? If the mathematical model is likely to be robust to quite a wide range of error, maybe you can actually reduce your experimental effort and make it easier for you to do your experiment because you do not gain much more from increasing the effort. Okay. Let's talk a little bit more about regression since we've been talking about ANOVA for a while. Now, regression is just the same model as an ANOVA. It's the same equation of y equal to alpha plus beta x plus epsilon. There's just some small differences in meaning. Y is still the response. X is no longer a 0 or 1, but some continuous value. Alpha is not the mean of one group, but it's the intercept. So where that line intercept, so the intercept in this plot. And beta is no longer the difference between groups. It is just a slope of this straight line. The error again is normally distributed, typically normally distributed with a mean of 0. So in order to simulate data for a regression, all you do is choose values for alpha, beta, and the standard deviation of epsilon, that's sigma. Instead of choosing a sample size per group, you choose the levels of x, the continuous values on the x-axis that you will use in the experiment. And otherwise, the process is the same. Simulate your data 1,000 times, fit your model, estimate alpha and beta, and then investigate how much variation there is in alpha and beta. And I'm sad to say that this did not start from the beginning, but it will restart soon. And yet again, here we have data with a true intercept and slope, and with randomly drawn noise added to all the data points, and then the regression refit to it. And then we have the distribution of estimated regression slopes here, with the true value indicated by the red line. So in addition to what I just said, in regression designs, you have one more choice, which is what will your experimental levels be? That is, where are you going to put your points? So to give you an example, I'm going to show you two designs here on the left. The x-axis goes from, in this case, from the 1 to 10. That is, the design space has a minimum of 1 and a maximum of 10. And in the uniform design, we've got 10 points here. We have one data point, that is one level at each integer. So we have 1, 2, 3, 4, 5 to 10. And we can have another one which we can call an extreme design, which in this case is an optimal design, where we have half the data points at value 1 and half the data points at 10. Now, one of these designs gives us a more precise estimate. After I show you the result, indicating which is more precise, I want you to think for a minute, type your answer in chat, but don't hit enter till I say. When I tell you to hit enter, everyone's going to hit enter together so you don't see each other's answers. All right? So here are simulations just like the one I showed you. Same number of data points, same amount, same noise, so same sigma value. But you can see this extreme design has a much narrower distribution. It provides much more precise estimates than the uniform design. Why do you think that is? Tell me, type your answer, think for a minute, type your answer. Don't hit enter yet. All right, hit enter. Let's see what you've got. So the sigma is the same in both the designs, but several of you have, and there's no different groups here. This is a slope of a linear regression. We've repeated this 1,000 times, so errors in single simulations shouldn't be a big issue. But several of you have got the right idea here, which is that The extreme points in a linear regression are disproportionately influential. Now, you can do this later yourself, but with R script number 5, you can run these simulations and you can manipulate these and test for yourself what the effects are. And now I'm gonna, I believe, hand over to Ravi.
Yeah. Can you guys see this? 1 second. 1 second. Yeah, can you see this? Perfect.
Yes.
So, you guys just saw that like the location of the points essentially makes a big difference in how precise your end result is, how precise your parameter estimates are, right? And some of you got the right idea as well, right? Like that you that I mean you sort of got the gist that not all points are essentially equally valuable along this entire sort of design space going from 1 to 10, right? And so, but how do we know what points are more valuable than the other? So there's this thing called leverage and in statistics, which sort of tells you how valuable a point is. And specifically here, I'm showing a version of it called generalized leverage, which essentially measures relative influence of a point on the eventual fit of the parameter estimates, right? So and how it does this is what it does is it combines what are called parameter sensitivities. So what is a parameter sensitivity? It is how sensitive is the function output to a change in the parameter. So, and it often depends on the predictor value. Now I'll give you an example. So we've been talking about a line, a regression, a linear regression, right? So far, a line, as you've seen the equation several times, right? There are two parameters in a line. There is a slope and there is an intercept. Now let's think about a slope. It's easy to think about to start with. So let's say you have a line y equals 2x, your slope is 2, right? And if you want to change the slope to, let's say, 5, how much is that going to change your output, your y, right? And of course, that will depend on the x value. So at x equals to 1, if you have y equals 2x, y equals 2. X equals to 1, if you have y equals 5x, y is 5. So there is a difference of 3. Same difference, if you measure at x equals 10, becomes much more. So the idea being that the parameter sensitivity increases in this case of the slope with the predictor. And each parameter has a different sensitivity, and we don't need to go into the math of all of these, but the intuition here is that the leverage combines each of these to tell you at any given point along this x-axis here, right, of the regression, where how much is the function, how sensitive is the function to a change in the parameter, right? And eventually that translates into how much influence does the point actually have on the fit of that parameter. And so this is a good diagnostic tool. So in this graph, you can see that this is a leverage for linear regression, and we just sort of saw this in the previous exercise. So you have the highest leverage values at the extreme ends, right? And so which is why when we in the simulation, when we put the points, all of the 5 points at 1 and 5 points at 10, you got the best estimates, and that's because these points have the highest information. Like they give you they give the regression the highest information. And but this is a good diagnostic tool. But how do we actually like develop a design? I mean, you can't. It's a line, so things are very simple here. You can actually just put points there at the end, and that works out. But doesn't always hold up in more complicated scenarios. So then we get into this whole new field of what is called optimal designs. And this is from like the design of experiments theory and statistics. And what you can do there is you can calculate the mathematically best design possible. And what is best essentially depends on 4 things. Okay. One is you have to define an objective of the experiment. And this sort of goes back to the very beginning of this workshop, which in you have to be clear about what the objective of your experiment is. So it can be something like you want the best parameter fits possible. It could be that you want some sort of predictive power. It could be a bunch of other stuff as well, right? But you have to be clear what the objective of your experiment is. Second thing you need is a function that you are going to fit. So in case of a linear regression, it's a line. If you're going to fit a monocurve for like nutrients versus growth, then that's the monocurve is the function, as we will see in a second, and so on. And you have to be sure that that function actually applies to your scenario, right? And then the other 3 and 4 are essentially things that you probably already know if you're doing an experiment. Like you have to have the range of predictors. So for example, if temperature is a predictor, you need to know what's the minimum temperature I can measure my growth rate at or whatever else at, and what's the maximum. And finally, you have to have the number of measurements. So let's say, how many measurements can I make? Is it 10, 15, 20, 30, whatever else it is, right? So these are the 4 things you need. Today, we will focus only on the reducing of parameter uncertainty, which is what we've been sort of going with so far. And I want to go back to the sort of the linear regression simulations that Mridul was just showing, right? And you can see here that I think he only showed maybe one of these, maybe the slope or the intercept. But essentially, these are both the parameters, the slope and the top and the intercept, right? And in the blue is the uniform designs simulation, and in the red is the optimal. And you can see there's a spread here, right? For both uniform and optimal. Let's focus on the uniform because it's more visible right now. And that spread essentially depends on, as you can already tell, it depends on the design. And that you can actually use some math, which I will not go into, to measure what that spread is for any given design, right? So how you can do this is this thing called a confidence ellipse. And so what you do is, let's say you have 2 parameters, so you put them on a graph. On x-axis is the intercept, on the y-axis is the slope. And this is our true value. The x is the true value that we actually did everything with. And then we take every single fit that makes up this histogram, and we put them on this graph, right? So it looks something like this, right? Now you can already see that there's some sort of like an ellipse here, right? And you can actually calculate using math a 95% confidence ellipse. This is slightly different from confidence. This is different from confidence intervals that you guys are probably used to. This is essentially saying, given a design, how much variability is going to be in your, given a design and a standard and an error level, how much variability is going to be in your parameter estimates. It's just the measure of the width of these distributions on this graph. It's the same thing, but a mathematical way of measuring this. So why I'm telling you all of this is because one of the things that you can do is you can use this thing called d-optimality criterion, which takes this ellipse, this is the same ellipse, and it minimizes it. So for any experiment that you are going to do, you can do this math and it will give you a design which will minimize the area of this ellipse, which is equivalent to maximizing the information content of the parameter estimates. It is giving you the most precise estimates possible, given your function, given your objective, given the number of measurements you're making, uh, and so on, right? Like, so given your conditions, it tells you the best possible design. So so there is that. So now you have already seen this, but just to sort of reinforce it, the optimal design for a linear regression is essentially you put, let's say we had 10 points, so five at one end and 5 at the other, right? And this, I already showed you the leverage. It matches the leverage quite well. Which, by the way, it doesn't always happen. It's the line. It's simple, so it works out this way, which is nice. But yeah, and then of course these are the simulation results that you already saw before as well, right? And then you can see this, and then you can plot like the blue ellipse is the one you saw earlier for a uniform design, and then the red one is the optimal, which is where the points are at the end, right? And you can see that the red ellipse is smaller in area than the blue one, right? So that's the gist of that. But what does this mean for you as a person doing experiments? It's all a bunch of nice math, but should you put all the points at the extremes if you are doing a linear regression, right? That does that, and we think the answer is actually no, right? This is nice math, but you have to think about other things as well. So the advantage, of course, is that if you do put them at the end, then you'll get the highest parameter precision possible, right? That's the nice bit. The not so nice bit is that you don't, if you put all the parameters at all the points at the ends, you don't know what's happening in the middle, right? If you are sure that it's a line that your system, like it is, that you're measuring is a line, everything's great. However, what if the function is nonlinear in the middle, right? And which, is quite possible. And in fact, actually, like, and we will talk about nonlinear regression next. And because almost nothing is actually linear in ecology, as you are probably familiar, things are almost always nonlinear, right? So with that, we will do another exercise. Mridul, you wanna walk
Sure.
this?
Sure.
Yes.
Can everyone just take a screenshot of that? Ravi, just keep it up there for a moment.
Yes. Yeah.
So your R script number 7 includes code for one kind of nonlinear regression, which is here we're going to simulate the mono function, which is what you see on the bottom right. This was devised for, it's roughly how bacteria respond to inorganic nutrients, but this is basically how any organism responds to food. You give the response is some saturating function of food availability. Now, in script number 7, we have coded this up and we have given it parameters for the two parameter values, the mu max, that's the maximum growth rate, and Ks, which is the half saturation constant. Now, what we want you to do is you have 16 data points. This is your budget. The minimum nutrient concentration you can do in your experiment is 0. The maximum is 15. You can spend your 16 data points anywhere between 0 and 15. If you want to put them all at 5, you could do that, though I wouldn't recommend it. But you could also do, you want to do like 3.2 and 14.3, you can do that as well. You have 16 data points. Your task is to come up with 2 different designs and then compare their precision. You just have to modify the top part of the script and then run through it and look at the histograms that we've just shown you. Go ahead and do that. 10 minutes.
Well, there we go. Okay. Well, I hope you got to try out a couple of designs and see how, and it might not have been like immediately intuitive as well. I can feel like to see how the design changes or the precision changes with the design, right? And the just to sort of, after you've done this exercise, just to give you the answer actually how to this puzzle. Basically, so this is again the same leverage plot, that the diagnostic that we used for linear regression, right? Which tells us how valuable our points are. And you can immediately see that there are sort of like these two high values, right? And why are those values, why those points, that means are the providing you the best information, and they're probably putting points at those measuring the growth rate at these points is going to give you the best parameter precision, right? That's the sort of the gist. And but why is that? So if you think about a monocurve, there are 2 parameters. Like actually, I can even go back. So there is the mu max, which is the maximum growth rate, which is essentially the growth rate at really high nutrient levels, and then there's the half saturation constant, which is essentially the nutrient value at which you get half of the maximum growth rate, right? So it's somewhere around here probably, and you can, you can sort of see that this point, because we, in practice, of course, we can't measure like infinitely high nutrient levels. So this point is giving you sort of information about the one at the extreme is giving you information about sort of mu max, which is that's the highest growth rate we can measure, and that is what is it's feeding into it's giving you most information about mu max. On the other hand, this is around the smaller point is around the half saturation constant, which was I think around five,
6.
six. Yes. Yeah. So it's a little bit before that, but it's around there. So which sort of gives you the most information about the half saturation constant, and you can sort of see it. So this is, by the way, if you do calculate the optimal design, it turns out to be around half of the points here and at the extreme, and half of the points at right before 5. Um, I don't know how many of you actually got to something close to this, but it's not like immediately obvious. So please don't worry if you didn't. And this sort of corresponds to in the curve, it corresponds to right about here, right? And you can do the same simulations, and that's sort of the gist here, right? Like you can do these simulations and you can actually try it out and see. And you can here, for example, we're comparing like a uniformly divided 16 points versus like the this specific design. And you can again see that the optimal design is, it does better. And you can do the simulations and you can check that out, right? That's sort of the gist. So, but all of this is great in theory, once again, but there are challenges specifically with nonlinear regressions. Okay. You will, you must have noticed that we gave you the parameter values and then we asked you to come up with a design, right? And that's because the optimal design for nonlinear regressions depends on the parameter values. Now, this is unfortunate because you might be like, Ravi, but I don't know the parameter values for the curve that I'm going to measure, right? So you sort of get into this catch-22, right? Which is the optimal design cannot be derived without the parameters, and the parameters cannot be derived without the optimal design, right? You get stuck in this loop. So, and so what happens if you calculate a design with the wrong set of parameters is one question. And are there solutions to this catch-22? The answer is yes, there are solutions to the catch-22. Um, and so which basically addresses this issue of the wrong set of parameters, right? And so I'm going to go over sort of two broad sets of solutions, and this gets complicated slightly mathematically. So please hang out afterwards, and we can answer questions if you have any about this. The first solution is what is called a pseudo Bayesian design. So you may not have the exact parameter value for the mono curve that you're measuring or for whatever else other sort of nonlinear regression that you're doing. But maybe other people have done similar experiments in other species, and you can get some sort of a maximum or a minimum, and you have some sort of, maybe you can construct some sort of a prior distribution based on biologically meaningful values and so on and so forth, right? What you can then do is you can draw one set of parameters from that distribution, right? So let's say one mu max and one k in this case. You can use that to come up with one design, and then you do this for another set, you get another design, you do this 10,000 times, let's say, and then you get 10,000 designs. And then you do some sort of averaging of these designs and put the points roughly on an average, right? That's one sort of, it's called pseudo Bayesian because there's a prior involved, but that's one way of doing this. The slightly better but way more mathematically complicated way of doing this is what is called fully Bayesian design. So in this case, what you do is you start with the design that you have, with the candidate design. So this could be anything. You can just start with like a uniformly distributed points, right? And then you once again need to have a prior, and then you draw from this prior like we did last time, right? But in this case, and then what you do is you draw from the prior and you use that to create artificial data that we've been doing in the simulations. You use that data to then do Bayesian fitting on this, right? So this is why it's called fully Bayesian. So you're now going to do Bayesian fitting on this data, and then you have a prior, and then now you will get a posterior, right? So this is posterior one. You can see how much better the posterior is with the prior, from the prior, using a set of utilities that I don't want to go over, but you can measure essentially the difference between the two. So now you get a measure of how good your posterior is. So that is utility one, right? Once again, you do this 10,000 times. Now you have, for the same design, you have 10,000 different utility values. You average them, you get one utility value for one design. You try this out for 10,000 designs, right? So what you're doing is you're sort of trying to see all the possibilities for all of these designs. What is the data I can generate? What is the posteriors? And then exactly which design would be the best at giving me the best average, the best posterior, essentially, really, right? That's what this utility is measuring. So then you have 10,000 of these utilities, and then you choose the best one and the design corresponding to the best one, really. So that's sort of the fully Bayesian route, right? And you don't have to do this. This is like a sort of still a developing thing, and we are still working out the kinks for it, and trying it out with different common functions. So once we have that sorted, we might uh, publish that, and then that would be the next step, essentially. So, do we have time for this, Mridul?
Anna, is it okay if we go like 5 minutes over?
Yeah, if this is the last thing, then I think we can do it. And then if folks have to leave, feel free to leave.
Let's say 8 minutes for this, and then we have just like a very brief wrap up. Okay.
Okay.
Now, if you look at R script number 8, we've given you a different function, which is what you see on the bottom right. So this is growth as a function of light. And here we want you now to find a good design for this function. There's 3 parameters. Again, there's mu max, the maximum growth rate. IOPT is the optimal irradiance. And alpha is the slope when the x value is really, really low. So the script already has the values in there. Now, what we want you to do is to think of a light range from 0 to 1000. You have 10 points that you can put anywhere from 0 to 1000. And come up with 2 designs and figure out which is better. First, do that individually, but after 5 minutes, we'll send you into small breakout rooms again so that you can compare your 2 designs with someone else.
Okay.
Okay, so I hope you were able to explore some different designs and look at their pros and cons. And depending on the designs you chose, you might have even found that some designs give you more precise estimates for one parameter, but less precise estimates for another parameter. So that's the kind of thing that the questions that you can start to explore with this approach. The other thing I wanted to note is that we've been talking about optimal design, but we're aware that it's complicated and also there are limitations there. So what we really want to stress is that you do not need to do an optimal design, but you can explore the consequences of many different designs and find things that match your objectives this way. So one thing we don't have the time to talk about today, but there are several extensions of this. Pretty much any model that you can think of, if you can write the model equation, it becomes easy to simulate and go through this exercise. Multiple regression, mixed models, GLMs, and non-Gaussian errors, very simple to do. So to conclude, again, defining a clear, achievable, useful objective is the most important thing you can do. Think about what parameter you really want to know and how precisely. Think about the model, that is the function that you're going to fit to your data, and the magnitude of the error. With this in mind, you can simulate data from all kinds of different designs, and you can compare their precision, and you can check robustness. What we didn't get to do today, but I really suggest you do, is figure out how well a design does if so Ravi has provided you an optimal design for mono. Think about how well that does if we actually had guessed the wrong parameter values. You might find that an optimal design would still do better even if you're a little bit wrong. That's what we found in our explorations. But those are things you can easily check with this approach. and with these scripts, hopefully. Again, you don't need to aim for optimality, but with simple simulations, I think we can all do better designs. And with that, we will draw this to a close. Sorry, we ran a few minutes over. And we will stick around to take any questions you might have.